As stated, my name is Kelvin Gordon, and today I'm just going to talk to you about our work here at the U of A and how we're able to kind of elucidate Remdesivir's mechanism of action against the SARS-CoV-2 polymerase and how this ultimately helped provide support for the FDA approval of Remdesivir as a treatment for COVID-19. So very briefly, kind of overview of what I want to talk about. I'll touch a little bit on nucleotide analogs and the development of Remdesivir as a broad spectrum antiviral. We'll go through our work and our lab's work with regards to the biochemical investigation of Remdesivir against different viral polymerases, including SARS-CoV-2. And then finally, we'll discuss a little bit Remdesivir as a treatment for COVID-19, kind of where those initial clinical trials put us at and where we sit now with regards to what we know and what's important when using antivirals for treatment. But to start off, I think a good place to begin is with regards to emerging viruses and the necessity or the need to continue developing different antiviral therapies. We can look back in history and get an idea of just the devastating toll that emerging viruses have had on the human population. We can go all the way back to 1918 with regards to the Spanish flu, responsible for approximately 50 million deaths. And as we move forward, we can see there have been a number of outbreaks and pandemics, including HIV, which is responsible for more than 30 million deaths. And as we come closer to present day, we see that there have been a couple of coronavirus outbreaks that have had a toll on human mortality. With regards to our lab and the focus of our lab, we really do it stems from ultimately this list produced by the World Health Organization in 2015, in which they determined a set of priority pathogens or diseases. These are emerging diseases that are likely to cause major epidemics and ones for which we currently lack any medical interventions. Included in this list are Crimean Congo hemorrhagic fever, Ebola virus and Marburg virus, as well as Lassa fever, Middle Eastern respiratory syndrome coronavirus or MERS-CoV, and severe acute respiratory syndrome coronavirus. There's Nipah, Rift Valley, Zika, and Disease X, obviously representing the unknown. I think we can appreciate this a little bit more now going through the pandemic and having to deal with SARS-CoV-2. I highlight in the middle MERS and SARS because I think this brings about the point that we did know the pathogenic potential coronaviruses possessed. And this was ultimately where my project began. So when we think about the development of antivirals, we have to consider the viral life cycle. And I've kept this picture, this diagram very general. Ultimately, viruses will infect our cells and generate progeny that are then released and move on to infect other cells. Now, when it comes to antiviral development, we need to consider the intricacies of the processes that are taking place during infection. And this way we can identify some potential therapeutic targets. These intricacies will differ from virus to virus, and this does make the development of therapeutics a bit of a black box. Fortunately, replication is something that is absolutely necessary for the infection of viruses. They require it in order to generate their progeny and move on. So, and this process is carried out by the viral polymerase. So you can think of the polymerase ultimately as the engine of that virus. And its necessity is what makes it the perfect target for antiviral drugs. It's also, with regards to viral polymerases, they are conserved amongst the RNA viruses, and they do differ from human polymerases enough that we can generate antivirals with enough specificity where they won't be used or used as a substrate by the human polymerase, so we can prevent any sort of potential off-target effects. Now, if we consider nucleotide analogs, and this is essentially my project, and broad spectrum nucleotide analogs specifically, they have served as cornerstones for antiviral development. We saw that early on with HIV and AZT, which is a nucleotide analog, and was the first approved treatment for HIV. And today they use tenofovir and emtricitabine, which are both nucleotide analogs commonly employed in the triple therapy approach against HIV. We've also seen success of nucleotide analogs against RNA viruses, such as hepatitis C, where the development of sofosbuvir in the late 2000s has proven to be very successful in preventing this treating this RNA viral infection. Now, a big question that kind of comes to mind for our group is, what about a nucleotide analog that can target more than one RNA virus? And this is an important question when considering pandemic preparedness, because we potentially are preparing for what would be unknown. And so it is more efficient to have a potential analog that can go after a number of different RNA viruses. And there has been some work in the field of broad spectrum nucleotide analogs. So we have up top here what we could consider our first generation. And so I show ribavirin and favipiravir. Both have shown an antiviral effect against different RNA viruses, although they didn't appear to be as effective against SARS-CoV-2, and they function as mutagens. Below is what we could consider our second generation of nucleotide analogs. On the left is remdesivir, and so this has received full FDA approval for the treatment of COVID-19. And on the right is molnupiravir, which has received emergency use authorization for the treatment of COVID-19 patients at risk for serious disease progression. Now, obviously, I'll focus on remdesivir today, and I think the story of remdesivir really begins with the Ebola outbreaks in 2014 and 2018. So the 2014 outbreak in West Africa of Ebola really prompted Gilead Sciences and collaborators to develop remdesivir as an antiviral treatment. With regards to the discovery route of remdesivir and kind of the processes that took place to ultimately generate what we know now as remdesivir, this collaboration was done with the CDC as well as the USAMRIID. And they ultimately screened a library of nucleosides and tides before identifying some potential hits that showed antiviral activity. And they came across what is considered a nucleoside GS441524. Now, in order to improve ultimately its the bioavailability, they have to create a prodrug in order to, like I said, to make it more effective. Now, the prodrug is important, especially when you consider the intracellular metabolic pathway of remdesivir. So remdesivir, like other nucleotide analogs, must be triphosphorylated in order for them to be incorporated by the viral polymerase. On the left here, we have the prodrug form of remdesivir. So this is what's delivered intravenously to COVID-19 patients. And as you can see, it has this prodrug moiety on the left, different from its nucleoside on the right. This prodrug moiety has a couple of important roles. The first being it allows for efficient bypass through that membrane bilayer. So we need obviously improved, or we need efficient cellular uptake for this drugs to have their effect. It also contains a single phosphate group already attached. And this is very important because during the phosphorylation process, it's the addition of that first phosphate group that is the rate limiting step. So if we can bypass this rate limiting step, we're ultimately creating or allowing for a more efficient process to take place, where ultimately the prodrug will become hydrolyzed and hijack essentially the endogenous phosphorylation pathway to generate the active nucleotide triphosphate form of remdesivir. And this is really what our lab will focus on in terms of understanding remdesivir in its active form and what that mechanism of inhibition looks like against SARS-CoV-2. Now, as I mentioned, remdesivir was initially developed as a potential antiviral for Ebola virus disease, and it did go into clinical trial, and it was tested or investigated alongside two monoclonal antibody therapies. Ultimately, the two antibody therapies proved to be more effective. That's not to say remdesivir wasn't effective. It just didn't reduce mortality to the same extent as those antibody therapies. But I would argue most importantly, this being the first trial with remdesivir allowed for the generation of human safety data, which ultimately became crucial with regards to moving forward with clinical trials against SARS-CoV-2. As I mentioned, remdesivir is a broad spectrum antiviral. So it's demonstrated antiviral activity against a number of different RNA viruses, and this includes coronaviruses. So as depicted here, we have EC50 values, or the effective concentration at which you're inhibiting 50% viral replication. And it shows a nice low EC50 value for the Ebola viruses, but it also shows potentially even lower values here for MERS and SARS, two coronaviruses. And this is really where kind of my journey began at the U of A. And so joining the lab in fall 2019, we had known about the clinical trial. Our group, in a publication led by Dr. Yegor Technikov, who's a research associate within our lab, has been absolutely instrumental in all the work that I'll show from our lab today, was able to demonstrate the mechanism of action of remdesivir against the Ebola polymerase. And this is important because it ultimately laid the foundation for my work in beginning to understand how remdesivir could potentially work against coronaviruses. So we decided in the fall that my project was going to focus on MERS-CoV, just following suit with the priority pathogens list as determined by the WHO. Now, by December 2019, we had some really nice preliminary data identifying a potential mechanism of action for remdesivir against the MERS polymerase. And we go away for Christmas break and we start to hear rumblings of this unknown respiratory virus in Wuhan, China. And by the time we come back, it's been identified as this novel coronavirus. And before the end of the month, the first case is identified in the United States. And so at the time, I think we were still very unsure of what this meant. But regardless, we saw it as an opportunity to help push the development of antivirals forward, and specifically focusing on remdesivir. Early on, we knew remdesivir was able to inhibit SARS-CoV-2 replication. And so Wang et al. Published the first study of the antiviral activity of remdesivir against a clinical isolate of SARS-CoV-2. And this was very important preclinical data with regards to moving forward for clinical trials. Now, by March 11th, the WHO had declared COVID-19 as a pandemic. And I think at this time, everyone was definitely in different situations. Our lab, we were fortunate enough to keep working, and we were trying our best to help understand just how some potential therapeutics like remdesivir could work against SARS-CoV-2. At the same time, there was quite a bit of data coming out, and it was definitely overwhelming and oftentimes could be contradicting. And the clinical trials are no exception to this. So we saw first a clinical trial come out that was led by the National Institute of Allergy and Infectious Disease, in which they showed that remdesivir was superior to placebo in shortening time to recovery. Another clinical trial comes out, and this was done by the WHO, in which they established that remdesivir, or they stated, I should say, remdesivir and other potential antivirals or therapeutics did not improve overall mortality. And so I think these contradictions point to some potential reasons for this is variations in study design, as well as a different patient population, timing of treatment, which I'll talk about later on, how important that is, and as well as endpoints. And I think it's important to consider that for both these clinical trials, they didn't necessarily determine the virological efficacy of remdesivir, and that they weren't measuring the amount of actual replicating virus present before and after remdesivir treatment. Before we get into that, we'll really focus on what our lab does here at the U of A, and we really want to understand how remdesivir works. So remdesivir is an ATP analog, so it will compete with ATP for incorporation by the viral polymerase. And this is a really nice depiction put together by Catherine Selye-Radke from UMBC, identifying the important features of remdesivir that allow it to elicit its antiviral activity. As I mentioned, there's that important first phosphate already attached with the protected by the prodrug moiety. It contains two nitrogens that allow it to act as proton donors and acceptors on base pairing with uracil. It has a glycosidic bond between the sugar and the base, and this helps improve the stability of the drug. Perhaps most important is this 1' cyano group that is ultimately responsible for remdesivir and what allows it to elicit its antiviral activity. And finally, it contains a 3' hydroxyl, and this means that once incorporated, it can be extended, and I'll talk about that later on. So from February 2020 to September 2020, we put out a few publications just trying to understand the biochemical parameters that allow remdesivir to elicit its antiviral activity. The February 2020 publication was specific to MERS coronavirus polymerase. This came out before the WHO announced the pandemic, and it was the first demonstration of the mechanism of action of remdesivir against a coronavirus polymerase. I think what's most important is it really gave us a foundation so we could move seamlessly into studying the polymerase from SARS-CoV-2, in which we ultimately determined that the three parameters are efficient incorporation, so how well is this nucleotide being utilized as a substrate by that polymerase. Next is delayed chain termination, or that first block in RNA synthesis. And the third is a second mechanism of action, template-dependent inhibition, or the second block of RNA synthesis. So if we first focus on efficient incorporation, what we are doing to determine how well remdesivir is incorporated is we will determine its efficiency of incorporation, and we will compare that efficiency to the natural counterpart ATP, and we come up with what we call a selectivity value. And so before I show the selectivity values, I just want to state that selectivity, when less than 1, indicates the nucleotide analog is incorporated more efficiently than its natural counterpart. So in this case, remdesivir more efficiently than ATP. When we look at the polymerase from different coronaviruses, we see that selectivity value falling around 0.3, indicating remdesivir is incorporated more efficiently than ATP, which is pretty amazing with regards to what we'd known previously with different nucleotide analogs. We wanted to take this study a little bit further and potentially understand what makes remdesivir better than other nucleotide analogs, and what structures might be important when considering nucleotide analog development. So we looked at some investigational compounds, and we found that for the most part, they weren't incorporated that well, all generating selectivity values greater than 100. But what's most interesting is we looked at favipiravir and ribavirin, a couple of broad spectrum nucleotide analogs I mentioned earlier, as well as sofosbuvir, the approved antiviral for the treatment of hepatitis C. And as you can see, the selectivity value is greater than 500, at least for all of them, indicating that the polymerase has a very difficult time incorporating these nucleotide analogs, suggesting that they're not going to be able to outcompete their natural counterpart. And this ultimately would suggest that they're not going to have an antiviral effect. Now, not long after that publication, a publication led by Z et al. Looked at different nucleotide analogs and their impact on SARS-CoV-2 in cell culture. And so we see remdesivir generates again a low EC50 value, generating that it's having a nice inhibitory effect, versus sofosbuvir, favipiravir, and ribavirin having an increased EC50 value. So if remdesivir really right now is serving as the benchmark in terms of antiviral development, we know based on these values that sofosbuvir, ribavirin, and favipiravir are not having a great antiviral effect. And it really shows a nice correlation or link between our biochemical data aligning with this in vitro data. Structurally, we can compare them and maybe make some assumptions as to what's happening and what's required. And as we can see here, when we look at sofosbuvir, the structures will differ significantly when you consider the 2' location. This is the modification responsible for inhibiting hepatitis C, whereas remdesivir is much different in that it has the 1' modification. Now, as I mentioned, remdesivir is a broad spectrum antiviral. And so it had shown antiviral activity against the coronaviruses, but as well hepatitis C, another positive sense RNA virus. It also showed antiviral activity against Ebola, RSV, and Nipah, all segmented negative sense viruses, but it appeared to be less effective in cell culture against Lassa, Crimean Congo, and flu, some segmented negative sense RNA viruses. And so in this undertaking, this involved work by myself, as well as Hedy Lee, who's a PhD student in the lab, and again, Dr. Yegor Teshnikov, in trying to understand what allows remdesivir to elicit or not elicit its antiviral activity. And so what we found is when we looked at the selectivity values, again, if selectivity is less than 1, that indicates your analog is incorporated more efficiently than its natural counterpart. And we see with the positive sense polymerases that it incorporates remdesivir at least as efficiently as ATP, correlating really nicely with the antiviral data. As we move on, we see the non-segmented negative sense polymerases, the selectivity value begins to climb, indicating that remdesivir might not be incorporated as well, but still it is incorporated and there is an antiviral effect observed. Now, where it gets really interesting is if you look at the selectivity value of these segmented negative sense polymerases, it really begins to climb. And this correlates really nicely with previous cell culture data indicating that there isn't an inhibitory effect. So the takeaway here is that efficient incorporation appears to be a prerequisite for inhibition. But this doesn't necessarily mean it's a sufficient requirement. So we want to understand next what's happening once remdesivir is incorporated. And so what we will do is we will incorporate remdesivir and we will monitor RNA synthesis beyond its incorporation. And what we find with the SARS-CoV-2 polymerase is it incorporates remdesivir and then it moves 3 nucleotides downstream. Now, this is possible because, as I mentioned, the remdesivir has a 3' hydroxyl group allowing for its extension. And at this I+3 location, it will ultimately stop RNA synthesis. Modeling suggested that there was a steric clash occurring between serine 861 and the cyano group of remdesivir. And when we mutated that serine, we determined that that was in fact true, and we're able to eliminate delayed chain termination. Now, after this study, a group led by Kochik et al. Did a structural examination of remdesivir and SARS-CoV-2 polymerase, and they identified, or they were able to actually visualize that steric clash occurring between the incorporated remdesivir and serine 861. So again, some really nice data aligning with our biochemical data. Finally, lastly is when you incorporate or when you increase nucleotide concentration, we're able to overcome delayed chain termination. And so in this instance, we now have a scenario where the viral polymerase of SARS-CoV-2 is able to synthesize a template that contains remdesivir embedded within it. So if you take a step back and broadly look at coronavirus replication, if remdesivir is present during the replication process of SARS-CoV-2, it's possible that those newly replicated genomes could contain remdesivir embedded within it. And our question was, well, what effect might this have on RNA synthesis? And so we wanted to look at the impact of RNA synthesis opposite unembedded remdesivir. And we note that the polymerase struggles to incorporate UTP opposite that remdesivir. So now we have a second mechanism of inhibition in which remdesivir is embedded in the template strand. And modeling suggests that there's a steric clash occurring with that 1' cyano group of remdesivir and the alanine 558 in motif F. Similar to delayed chain termination, we are able to overcome this mode of inhibition by increasing nucleotide concentrations. And this is important because when we're increasing nucleotide concentrations, we're increasing them to ones that might be more representative of what's observed at a cellular level. But in this case, the potency of this mechanism appears to be much stronger in that it requires a higher nucleotide concentration to overcome that inhibition. Now, in terms of our impact of our research for COVID-19 patients, so our work was included in the FDA fact sheet for healthcare providers with regards to the approval of COVID-19 for the treatment of approval of remdesivir, for the treatment of COVID-19. And this allowed Gilead to create training modules for clinicians so they could accurately illustrate to their patients just how remdesivir is working. So hopefully this would help decrease any sort of patient hesitancy, but also help facilitate more positive patient-doctor relationships. And to date, remdesivir has been administered to over 12 million patients worldwide. Now, as I mentioned, there is some mixed clinical data when it comes to the efficacy of remdesivir against SARS-CoV-2. And I think it's really important to consider timing of infection. So early on in infection, we will see that the viral load will essentially increase. This is our replicating virus. And as time passes, the viral load decreases, but we now see this inflammatory response, right? And it's this inflammatory response that we're seeing in ICU patients. And so what I want to note here is because remdesivir can only be delivered right now intravenously, it's very difficult to give in an outpatient setting. So essentially, by the time it's delivered to the patient, we've seen the infection kind of already take hold and have its inflammatory response that way. Now, I think a study that kind of takes a little bit of an interesting look at this idea was by Buckland et al., where they took more of a reductionist approach, where you have a patient who lacks the ability to produce an antibody response, right? So essentially, in this case, there won't be that inflammatory effect. And so what happens is when they treat the patient with remdesivir, they note that the viral load of SARS-CoV-2 is decreasing. It eventually rebounds before decreasing again with a second dose. Now, there was a clinical trial kind of investigating this question a little bit further and kind of what is the effect of remdesivir in more of an outpatient setting. And so what happened in this clinical trial was a 3-day course treatment of remdesivir was given to COVID-19 patients that were at risk for the development of severe COVID-19. And they found that it reduced hospitalization or death by 87%, the risk of hospitalization or death. And so again, I think altogether, this really highlights how important timing is when it comes to the treatment and using antivirals for the treatment of an infection. And also, as I mentioned, remdesivir right now is delivered intravenously, and this really is a drawback of the treatment, and because it is difficult to give in an outpatient setting that way. But there is work in progress in terms of trying to develop an orally available version of remdesivir. And so this is some preclinical data showing that the orally available prodrug of remdesivir is effective against SARS-CoV-2 in mice. And I do believe now that this orally available version is currently in clinical trials, so that could substantially help with regards to more efficient delivery to patient. Now, finally, I want to talk about some of resistance work that we've done in the lab. And so this was done in collaboration with Dr. Mark Dennison out of Vanderbilt University. And the biochemical data shown in this publication was spearheaded by Hedy Lee, the PhD student in our lab. And ultimately, what was done was Mark Dennison and his group passaged SARS-CoV-2 and increasing concentrations of remdesivir to identify some potentially resistant mutations. There were 2 really interesting mutations identified in the polymerase. The first is the serine at position 759 to an alanine. And this is the active site of the polymerase, so the region that's responsible for incorporating the nucleotide. And what we found biochemically was this mutant actually, instead of having a preference for remdesivir, has a preference for ATP. Another interesting mutation was valine 792 to isoleucine. And in this case, we saw that this mutation allowed the polymerase to incorporate UTP opposite remdesivir more efficiently. So this would be a mechanism of resistance to that template-dependent mechanism. Now, after that publication, there was a patient who underwent an organ transplant and ultimately developed resistance to remdesivir, or was infected with SARS-CoV-2 and developed resistance to remdesivir. And the resistant mutation was that valine 792I. So a really nice connection from our biochemical and cell culture data with Denison's group and some clinical data. Now, I want to spend just a little bit of time talking about another broad spectrum nucleotide analog that has received emergency use authorization for the treatment of SARS-CoV-2, and that's molnupiravir. Molnupiravir is orally available, and so that obviously it has a leg up on remdesivir in that sense. And it has shown antiviral activity against other RNA viruses, including influenza. And at the onset of the pandemic, molnupiravir was developed by Merck and Ridgeback Therapeutics to treat COVID-19. So, and I think there's some interesting information that came from this. We saw, this was in July 2021, that the US government was giving some significant support to molnupiravir in its development. And I think that's because at this point, we really started to learn how important timing of treatment is and how important it is that we have an orally available version or an orally available antiviral for the treatment of SARS-CoV-2. Now, our group was able to illustrate the mechanism of action of molnupiravir against the SARS-CoV-2 polymerase, and it functions by way of lethal mutagenesis. So it essentially is confusing that polymerase to make mismatches in base pairing that ultimately overwhelm that viral genome, so infection can no longer occur. And so what we found is the mismatches that it promotes are these G to A and C to U transitions. Now, what's pretty cool is not too long ago, samples were collected from a clinical trial in which these patients who were positive for COVID-19 were treated with molnupiravir. They collected the SARS-CoV-2 samples from these patients, and based on these clinical data, they were able to see that there was an increase in G to A and C to U transitions. So again, a really nice connection between our biochemical data and later clinical data. Now, as I mentioned, molnupiravir is a mutagen, and so it does have its own drawbacks in that sense. And there was a really nice review put out by Swanstrom and Shinazi, just kind of highlighting maybe some drawbacks of using lethal mutagenesis as an antiviral strategy. So as I mentioned, these nucleotide analogs must be triphosphorylated in order for them to be incorporated by the polymerase. Before they're triphosphorylated, they are diphosphorylated. So there is the potential they could enter a pathway in which the enzyme ribonucleotide reductase, or RNR, is able to ultimately produce a potential substrate for the synthesis of cellular DNA. So it's something to keep in mind when developing these antivirals. I should note that a lot of the cellular data around molnupiravir doesn't necessarily suggest this is the case. So in summary and outlook, I'm just kind of going over what the past few years have looked like for our lab. We start really with those Ebola virus outbreaks, and our group was able to demonstrate the mechanism of action of remdesivir against Ebola, and this really was kind of our pandemic preparedness phase, if you will. Then early on in 2020, we were able to publish the mechanism of action of remdesivir against MERS coronavirus polymerase, the first demonstration of remdesivir against a coronavirus polymerase. And this was before the WHO had announced the pandemic. Not long after that, we showed the mechanism of action against SARS-CoV-2, as well as a second mechanism, and this helped support the FDA approval of remdesivir. We identified the broad spectrum activity of remdesivir and kind of what's required for it to elicit its antiviral activity against a number of different RNA viruses, as well as some potential mechanisms of resistance. So now as we move forward, we're really in this disease management phase. And so we've seen since the onset of the pandemic, a couple outbreaks occur, and one being the Marburg virus, as well as Langya, and of course, monkeypox is included there too. And so the question is, how do we prepare for a potentially unknown pathogen? And what our lab has done to kind of make strides towards this is we've established a library of viral polymerases that gives us a really great representation from important viral families. So should this unknown pathogen pop up, it's likely to fall into one of these families. So hopefully we have an assay in place and maybe an idea of what compounds may be more or less effective against that polymerase, hopefully helping to facilitate the therapeutic development. With that, I would just like to thank everyone on the Remdesivir team in our lab with Matthias Gott, Yegor Teshnikov, Dana Koknikova, Emma Wollner, and Hedy Lee, our collaborators at Gilead, Vanderbilt University, and University of North Carolina, and as well as our funding agencies that allow us to do our research. And with that, I will take on any questions.